So then I'll just press my share screen and go ahead.
Okay. Welcome everybody to the Cannabis Chemistry Journey Club meeting for February. I'm Matthew. I'm the Cannabis Chemistry Journal Club coordinator. We are fortunate enough to have with us today Julie Fry. Julie Fry is a part-time scientific consultant at the Plant Consulting Group. She's also currently enrolled at the how do you pronounce that? Masaryk?
Masaryk.
At the Masaryk University as a PhD student studying the biotransformation of cannabinoids. So, feel free to take it away, Julie. Allow me to unmute. Allow me to mute everybody really quick, and then.
Okay. Okay.
We'll go ahead and 1 second, please. Okay. Take it away.
Okay. So let me just
Yes, of course. No problem.
Okay. Let's get back a few slides. All right. So today I am presenting the structure and function of delta-1 tetrahydrocannabinolic acid, THCA synthase, which is the enzyme that controls the psychoactivity of cannabis sativa. This group has already published a paper several years prior to this one where they actually formed the first crystal of THCA synthase. So this group's been working on this particular enzyme for quite some time. And I will start off with, sorry, Matt, how do I remove the, do I just go like that? Yeah, there we go. Okay, just gonna put that down there. Okay, so what does THCA synthase synthesize? Well, obviously it synthesizes THCA because if it's responsible for the psychoactivity of the plant, then it would have to synthesize the precursor to its psychoactive component. So, and that is THCA, tetrahydrocannabinolic acid. Other byproducts that it synthesizes are cannabidiolic acid and cannabichromenic acid. And it does this through a hydride transfer and a deprotonation. And we'll find out how that actually occurs in the plant, through this group's work. They investigate the structure-function relationship of THCA synthase with the substrate. The substrate, of course, is cannabidiolic acid, which is the precursor to THCA and cannabis sativa L. And the method that they used was first to overproduce these proteins in insect cells. They then purified and crystallized the proteins. They bombarded them with X-rays by using a technique called X-ray diffraction. They analyzed the data, and based on the data analysis, they then decided to mutate ionizable amino acids, which they thought may be involved in this reaction. They then measured the enzymatic activity through reverse phase HPLC, and they compared the results to see which they thought were the most putative candidates for not only substrate binding, but also for the catalytic reaction itself. So how did they set up their experiment based on what information? Well, first they realized that delta-1 THCA synthase has a 39% homogeneity to BBE, which is berberine bridge enzyme, and a 26% homogeneity to glucooligosaccharide oxidase, and a 21% homogeneity to aclacinomycin oxidoreductase. These are various enzymes found in other plants that are responsible for pathogen response. And similarly, THCA synthase is the enzyme in cannabis that is responsible for pathogen response. And all of these enzymes are what are called flavoproteins, which means that they're coenzymes, and by the co signifies that they need a cofactor in order to complete their catalytic activity. What is the cofactor? Flavin adenine dinucleotide. So this moiety is essential for enzymatic activity, and it's essential for enzymatic activity in BBE, and therefore thought to be essential for enzymatic activity in THCA synthase. So they wanted to figure out, all right, well then, which amino acids in THCA synthase are responsible for binding the FAD moiety? And are they the same amino acids responsible for the catalytic activity of the enzyme, the hydride transfer, the deprotonation of CBGA? What's going on in there exactly? What's going on in that active site pocket? And so first they started out, as I mentioned, with protein overexpression and purification. This process is pretty straightforward. Essentially, this AFP here would be the THCA synthase. They would clone it onto a plasmid, then they would transform it into the insect cell. In their case, they used insect cells. They'll then culture these insect cells to create a massive amount of insect cells. They'll pellet the cells down with centrifugal force. They'll lyse the cells. They'll have the protein. They'll filter it out. They'll wash, and finally elute. And you have the purified protein. Now you can go for the crystallization. They used a combination of sodium citrate, pH 7.8, and 0.1 Heaps buffer. Actually, might have been, yes, so pH 7.8 of the overall mother solution. And this, in the end, was the tertiary structure that they came up with. And you can find all of these images in the paper. I just pulled the images from the paper so that I could explain them to everyone, just in case you didn't read the descriptions or whatever, for whatever reason. And so the tertiary structure of THC synthase includes beta sheets, which are in magenta, and alpha helices, which are in cyan, and these pink loop structures. And it has two primary domains, which are separated by this broken line here. And it has two subdomains in domain 1, which are separated by the dotted line. And they comprise different components of the tertiary structure. And the FAD is the binding pocket of FAD actually kind of straddles all three domains, but excuse me, it is covalently bound to amino acid residues in domain 1 primarily. And this, I couldn't find a difference between the two structures, but you'll see here FAD is a stick figure in blue in the very center of this crystallized structure. This is putatively what it looks like. So now, what does FAD look like? Okay, so they've done an electron density map where they merged the observed Fourier transformation with the calculated transformation, and this is the putative electron density map, and you can see that it's this area is these are the electrons. They're in cyan, and these two residues, histidine 114 and cysteine 176, are the two residues that covalently bind FAD to THCA synthase. Now that they figured out that these residues, so let me back it up just a little bit, and I'll tell you how they arrived at this enzymatic assay. So, excuse me. Okay. So basically, they were looking at several residues as putative amino acids that bind the FAD residue to THCA synthase, and that may also be responsible for the catalytic reaction. And if you read the paper, they list glycine 122, glycine 113, histidine 114, tyrosine 175, glycine 180, glycine 183, and histidine 184, asparagine 483, tyrosine 190, and tyrosine 481. So that's a lot of residues to be looking at. So they had to figure out, okay, well, which one of those may be involved? And in the first place, they realized by looking at the berberine bridge enzyme and superimposing the tertiary structures based on where the isoaloxazine ring of the FAD moiety is located, that in berberine bridge enzyme, there were a few amino acids that coincided with similar amino acids in THCA synthase, And those amino acids in the berberine bridge enzyme were responsible for the catalytic activity. So they thought, okay, well, we're going to investigate the analogous ones in THCA synthase and see if those ones are also responsible. And so that's how they essentially arrived at which amino acids to investigate. And once they figured out which ones were overlaid and putative amino acids either responsible for substrate binding, for binding to the FAD moiety, or for the catalytic activity of the oxidocyclization of CBGA to THCA, they mutated these residues. Okay, so histidine 192 was mutated to alanine. This was done by random mutagenesis. It's pretty simple in a laboratory. All you do is you create primers. So you target the mutagenesis to a specific region of the gene. The particular tertiary structure that they investigated, they used the primary structure. They used, I believe it was 545 amino acids and only 508 out of that to create the tertiary structure. But they take these amino acids, they put a primer in that directs mutagenesis to that particular site. And that's how they mutated all of these amino acid residues. So they mutated the histidine 292 to alanine, the tyrosine 417 to phenylalanine, the glutamic acid 442 to glutamine, tyrosine 484 to phenylalanine, histidine 114 to alanine. And they then looked at how the enzymatic activity of these mutated THCA synthases worked and they investigated that using reverse phase chromatography. So essentially what that means is that they incubated these mutated enzymes with the substrate, a substrate analog, and the product. The substrate is CBGA. The substrate analog that they use is cannabidioloric acid, and the product, of course, is THCA. And they're looking for the reaction. They're looking for, is it going to catalyze a reaction? Well, in wild type, of course, we would expect 100% enzymatic activity. So that's why it stays the same, right? Well, histidine to alanine, it lost 95% of its enzymatic activity. So that must mean that this is partially responsible for the catalytic reaction, if not entirely, right? Well, it can't be entirely responsible because you also see similar reactions here with the tyrosine mutated to phenylalanine and histidine 114 mutated to alanine. They both, well, this one, they both lost complete enzymatic activity. Whereas the tyrosine 417 and glutamine 442 only partially lost enzymatic activity, but they did lose enzymatic activity. So that means they still play some kind of role in the oxidocyclization of CBGA. So the reason why they investigated these specific residues, as I mentioned, is because overlaying the BBE, GOX, and ONC-OX enzymes, they noticed that those residues are located in the same active cleft pocket as the residues responsible for catalytic activity in those enzymes. And they did this by overlaying the enzymes based on this N5 position of the isoalloxazine ring of the FAD moiety, because remember, they are all flavoproteins, so they all bind to FAD moieties. And so they used this as the target for the overlay and found that these residues were all within 15 angstroms of this particular nitrogen of the FAD moiety. So they thought, okay, well, this is the active binding site cleft pocket, so they may have a role in the catalytic activity of the THCA synthase. Okay, so, and by the way, just to explain this view, this is a top-down view of the binding pocket as if someone were to slice open the THCA synthase enzyme so that you could see inside, in case you're wondering. All right, so now if you take away that substrate binding pocket and you just look at the proximity of these amino acids to the FAD moiety, you see that the tyrosine 484 is 3.4 angstroms away. The tyrosine 175, I believe, or excuse me, is that the tyrosine 417 is 7.1 angstroms, and the histidine 292 is approximately 10.5 angstroms. They only gave 3 measurements, but you can see approximately that they're all within 15 angstroms, so they could potentially have the ability to catalyze this reaction. And then again, you can see the pink moieties of the cysteine 176 and the histidine 114 covalently bound to the FAD, holding it in place so that it can go through the catalytic reaction of converting CBGA to THCA. Now you see this same stick figure diagram with the amino acids of the berberine, of the BBE, GOX, and OncoX enzymes overlaid. And you see what amino acid residues those ones particularly contained that made the researchers think that the comparable amino acids in THCA synthase might be responsible for this hydride transfer and deprotonation, which is hence why they chose to investigate these particular amino acids in the first place. And so finally, they determined that essentially, okay, let me just back it up a little and pause because I jumped ahead of myself, excuse me. So what they figured out was that the hydroxyl groups of the tyrosine 417 and tyrosine 354 are directed in the opposite direction of the FAD moiety. So they determined that they're probably not responsible for the deprotonation or the hydride transfer. However, they did determine that the glutamine 442 corresponds to the catalytic residue of the glutamine 417 of BBE. However, as you saw, and let me jump a couple slides back, as you saw in the mutagenesis assay, the glutamine 440 or the glutamic acid 442 to glutamine the mutagenesis did not result in a drop in enzymatic activity. So that one was probably not responsible for the catalytic activity of the enzyme. So what next? What residues could they investigate next? Okay, well, the tyrosine 429 and tyrosine 450 of the GOX and OncoX respectively are located near the tyrosine 484 of the THCA synthase. So they thought, okay, well, then this must be then the most likely residue that's responsible for the deprotonation activity of the oxidocyclization of CBGA to THCA synthase. And they also realized that they lost some enzymatic activity by mutating the histidine 292, but that it didn't seem to be responsible for the entire catalytic activity, but that when it wasn't bound, the enzyme would not go through catalysis. So they realized that it's probable that it functions as a counterion to CBGA carboxylate because, and the way that they investigated this was they tried to bind CBGA, or excuse me, CBG without the carboxyl group, and it wouldn't bind. So they figured out that the carboxyl group of CBGA is necessary to bind to the histidine 292 to hold the substrate in place while the catalytic reaction goes on. However, it does not play a direct role in the catalysis. And similarly, they determined that the tyrosine 417 plays a similar role in substrate binding, but not necessarily the catalytic reaction. So, in conclusion, here you see the tyrosine 484 coming in, going through the hydride transfer. Then you've got the deprotonation, then the hydride transfer, excuse me, the hydride transfer, and then you've got the whole cyclization reaction that essentially occurs because all of these bonds shift. And then you see here on the outside that the histidine 292 is in fact bound to the carboxyl group, and the tyrosine 417 is bound to this hydroxyl group here, but that these two groups remain the same. in the end, which means that they're unchanged. There's no active catalytic reaction going on here. The only reaction that's actually happening is going on with the tyrosine 484 and the FAD moiety itself. And here, as I mentioned, are the histidine 114 and cysteine 176 residues that are bound to, that are part of THCA synthase bound to the FAD moiety to aid with the catalytic reaction as well. And if you look then at the crystal data to determine, okay, well, how accurate is this study? They actually have a huge redundancy. They did 24,099 reflections, which is a very good amount of reflections. The discrepancy between their R work and R free, the R factors, is pretty low. It's not as low as you'd maybe want it to be, so they still may have overfit some places, but overall, this is a pretty solid crystal structure evaluation. And in conclusion, the FAD coenzyme covalently binds to the histidine 114 and cysteine 176 of the THCA synthase in domain 1, and it straddles subdomains 1A and 1B. The catalysis reaction goes through the hydride transfer from the C3 of CBGA to the N5 of FAD and the deprotonation of the O6' of CBGA. And the hydroxyl group of the tyrosine 484 is heavily involved in this deprotonation catalytic activity. And again, the histidine 292 and tyrosine 417 play a role in substrate binding, but little, if any, direct catalytic activity. That's my presentation. So are you still with me?
Awesome, awesome. You can go ahead and hit the stop share. Yeah, there we go. And if anybody has any questions, feel free to, you can either type them into the comments section, or you can feel free to unmute yourself. Yeah, feel free to leave a question in the comments section if you need to, also like in the message section, and I'll read it too.
Oh, okay, I was gonna say I don't see the message section.
Yep.
So I know that's a very heavy paper. I don't know how many people are familiar with biochemistry techniques, but I'm more than happy. I know I went through that kind of fast too. I'm more than happy though to explain anything that I may have kind of just skimmed over, if you have questions about any of that.
Anyone?
I just had a question here. Greg from Montreal. And can everyone hear me?
Yes, we can hear you.
Yes.
Oh, yes. I was just wondering if there was any studies that were done that look at the how we influence the THCA synthase to produce more. Were there any metabolomic studies done?
Good question. Well, since CBGA is the precursor, I would imagine if you produce more olivetolic acid, or I think it's olivetolic acid and cannabidrolic acid, because they combine and create CBGA, right? Or is it gerolic acid? I can't remember the exact precursors, but I would imagine if those two to combine more CBGA, you could then theoretically create more THCA, right? But it also has the other byproducts, the CBD and the CBC. So you would have to specifically probably then upregulate the catalytic reaction that we just spoke about with these residues. So that's a really great question. But I think that was part of the purpose of this paper, was to determine what the mechanistic reaction was, so that you could regulate the production of THCA synthase, or THCA, excuse me.
Very cool. Thank you very much.
Yeah, definitely.
So I have a question, Richard. You ready?
Yes, we can. Yeah, go ahead.
So when I look at the drawings of CBGA, CBDA, and THCA, I see one has one ring, the other one has two rings, and THCA has three rings. And just looking at the drawing, I said, oh, it must go from one to the next to the next.
Mm-hmm.
All this discussion is so very specific about THC and maybe all separate from CBD. Am I thinking correctly?
So are you saying that you thought that it would go from from THC to, or from CBC to CBD to THC? Is that what you mean?
Yes, adding one ring each time.
Mm-hmm. Actually, that's a great question, and they addressed that in the previous paper. They actually showed that counterintuitively, it doesn't. THCA isn't derived from CBD. It's a direct byproduct from CBGA.
Okay.
It's really, really interesting. I can send you the paper, the prior paper where they determined that using another crystallization study.
I'd heard it was 2 different pathways, but I just wanted to clear up that it's not just getting over the energy of producing one ring and then another enzyme comes along and gets around the energy of producing another ring.
Yeah, it's different pathways. It's really interesting. It's different pathways.
Thank you.
Yeah, definitely.
Okay, if anybody, any other questions? Feel free to unmute yourself. We have about 10 minutes still.
Yeah, hi, Julie Sanford Wolgell. Very nice presentation and explanation of paper. In terms of your question to Richard, what reference were you referring to about this alternate pathway? Is it in the paper?
So,
Is it in the paper? Is it one of the references in the paper?
it's in this group. Let me just see if I can find the paper. I have it on my desktop right now, actually. So this group, as I mentioned, they've been studying the crystallized structure of THCA synthase for quite some time. And they previously published a paper. It is called the crystallization of delta-1 tetrahydrocannabinolic acid, THCA synthase from cannabis sativa. It was published in Structure Biology and Crystallization Communications. And I'll send that. I'll send it to Matt. Should I send it to you, Matt, and then you can send it to everyone?
Yes, feel free if everybody wants. And then also in the comment section, I left my email right there. So if anybody needs to get in contact, or if anybody has any questions or anything that need to be passed on, feel free to don't hesitate to contact me, and I'll be sure to relay everything in between.
And do you mind? I'm going to share my screen just for 1 second. I'm going to show this reaction that I'm talking about.
Yeah, go ahead. We still have 8 minutes, so.
I have one more question too.
Okay, so this is the specific diagram where they show that CBD, it doesn't go from CBG to CBDA to THC. It's actually a direct catalytic reaction from CBG to THCA. Yeah, and I will send this paper to everyone, to Matt, and then he can send it to everyone.
Yeah.
And so that's.
So Julie, you certainly know all your mechanisms. You spent a lot of time on this, and it shows. So the question I have is last October there was a paper published by a group with GW Pharma looking at THCV and its potential effects on diabetes.
Mm-hmm.
And so the question, my understanding is that this same enzyme can also work on THCV. Is that the case here as well, or how does that work?
I don't know the answer to your question, but I will be sure to find out.
It's interesting because, I mean, when you look at this thing, I believe they said that this enzyme can work on THCV, which means that the business end of the molecule is not related to the either the propyl or the propane either 3-carbon or 5-carbon side chain. So, which means it sort of sticks out somewhere else.
Right.
It's pretty sloppy with respect to that. But I'm just curious about that.
No, that's an excellent question. That is a really excellent question. I have to look it up. I don't want to give you an incorrect answer. I don't want to just guess at it.
I'm just wondering because they don't mention any of that here, but I thought that that was the case.
Yeah, they don't mention THCV at all in the paper. But I like I said, I don't want to give you an incorrect answer, but it is really interesting. You're probably right that the side chain may have very little influence over the catalytic activity. It's possible. It's definitely possible. Yeah.
Okay. Thanks.
Yeah. Absolutely.
Yes,
Wish I could
you
help
are.
more. I will look it up, and if it's okay, I'll email you, or I'll email Matt the answer, and then he can shoot it off to you.
Yeah, I'd be curious.
Yeah.
Curious. Thank you.
Yeah, no problem.
Okay. Anybody else have a question or a comment?
One last question for me. I'm not sure if it was covered or not, but was there any studies done on if the precursor to THCA, CBGN, I believe, can be converted to THCA without the help of the enzyme?
Right. So that's a really good question too. Do you mean like in vitro or in vivo, or how do you mean that? Like could it be done artificially?
You know what? Both in vivo during the plant's life, for example, and maybe afterwards. Say there's a lot of CBGN that was produced, or CBN, sorry, in the plant that was still not yet converted. Is there a way to, without an enzyme, kind of like spray it or dunk it in some sort of solution and convert leftover precursor
CBGA?
to the precursor to THCA?
You know what? Actually, I'm certain of it. I'm certain of it because you could manipulate the mRNA or mRNA. So you could probably just skip that step entirely if you were to manipulate the genome to directly transcribe THCA. Yeah, you could there's ways to do it. I don't know that it would be organic, but yes, you could absolutely do it artificially. Do you know what I mean by manipulating the genome so that you would bypass that step and transcribe specifically THCA?
Oh, absolutely. I have a background in biochemistry and molecular biology. Yes.
There you go.
Thank you.
Go. Yeah. So you got me.
Thank you very much.
Yeah, no problem.
Nice. Okay, I think we'll have to call it quits here. We're actually down to about 3 minutes. I want to thank every single person for actually participating in the February Cannabis Chemistry Journal Club meeting. I want to thank Julie Fry for producing such an awesome presentation. I definitely want to thank you for that. If anybody is interested in presenting for any future papers, or we usually present every third Sunday of the month, but we're happy to reaccommodate. Please feel free to get in contact with me at matrocity818@gmail.com. I left it in the conversation section, so you can just copy and paste it if you need to really quick. If anybody has any questions for Julie, or if any of the links or anything, also feel free to contact me. I'll be happy to relay them. And feel free to tune in next month. I actually believe we'll be having James Yeary, another CAN member, and I believe he's also like an extraction technician and engineer. He'll be offering his kind of unique insights on developmental extraction methodologies in the cannabis industry. And I believe next month we will be having a meeting kind of in line with the ACS national meeting coming up in San Francisco. So feel free to tune in for that one too. That one's also going to be a good one too. I'm still planning to line up the speakers, so I'll be sure to inform everybody next month. And yeah, I believe that should be it. Again, thank you everybody for participating in the February CAN Journal Club meeting. Hope to see you next time, and everybody have an awesome weekend too. Thank you again. We really
Thank you, Matt.
Thank you.
Thank you.
Nice job, Julie. Thank you very much.
Thank you. Thank you so much.
have a good one, you guys.
Thank you.
Take care. Enjoy the day.